Okay, so today I'm going to be talking about some of the work that we do on what I call genomic dissection of trait complexity. And we really work quite heavily with commercial breeding companies. What you can see here on these two pictures are commercial canola, winter canola, and commercial sorghum breeding plots belong to two companies who we collaborate with. And really, what we do is try and support breeders to try and find or develop more efficient ways to select for complex traits. And this involves really understanding those traits and trying to find simpler ways than phenotyping or simpler ways than field phenotyping in order to select for these complex traits. I have quite a big group working for me to look at all sorts of different aspects of different crops. We work mainly on canola, but also on sorghum, on winter cereals, wheat and barley especially. And the examples that I'll give today are actually from sorghum and wheat. But really the methods that we're using are applicable to different crops, because in all crops you're faced with this dilemma that really the most important traits that we want to select for are complex traits right through to yield, which is the most complex of all, where we need really to simplify methods in order to select more accurately and more effectively. So these are the people who work for me. I have quite a few postdoc level or associate level people, a large group of PhD students, most of whom are excellent people, and I'll be presenting a lot of the work that they've been involved in, and you'll see the names at the bottom here, so I won't acknowledge individually. And quite a few technical people involved in molecular genetics work, quality analysis from seed and plant products, but also field analysis and field typing, so phenotyping. So we have 3 different experimental farms with quite different environments where we can do field phenotyping for diverse traits. And we also collaborate very closely with breeding companies who often do a lot of the phenotyping themselves in order to really get a big basis of data from many, many different environments. So some of the companies named here, there's a few collaboration partners named here, including, of course, the University of Queensland. So we collaborate with many breeders, many people worldwide to do all sorts of different interesting things. So the traits that we're really most interested in are anything that's not simple, more or less, but is of importance to a breeder. So we're looking at things like grain or biomass yield, and especially the mechanisms of heterosis and the ability of hybrid crops to improve their yield. We're looking at some very important quantitative disease resistances in some of these crops. Germination and seed vigor is something which I'll focus on today because it gives a nice example of the methods I want to describe. Seed and plant quality, flowering time is really important because flowering regulators seem to have a very really early and deep influence on many, many different complex traits through pleiotropism. And also more recently, adaptation to climate change and abiotic stress. This isn't just an issue here in Australia where the extreme climate is much more common, but even in Europe we're starting to have troubles with erratic climates, erratic climatic events, and breeders who are interested in pushing their products and varieties into Eastern Europe are really having to deal especially with drought and this kind of abiotic stress. Very important for European agriculture is nutrient use efficiency, so we're facing much stronger restrictions on inputs in agriculture, which mean that we really have to focus, especially in canola, for example, on nitrogen use efficiency. The approach that my group uses to try and dissect and understand these traits is an omics-based analysis, I call it. So it's basically using all sorts of different kinds of omics technologies, which I'll go into a little bit more soon. and trying to dissect a huge quantity of data to find the things which are most relevant for the traits of interest to us. So what we would like to do would be to identify genes if possible, or if not, then at least the networks which are underlying difficult traits. If possible, to develop kinds of biomarkers. They may be genes, they may be gene expression levels, they may be metabolite expression levels, or even models if things are a bit more complex, to enable us to more efficiently and accurately select for specific traits independently of environmental effects or constraints. So we're really looking for the genetic effects underlying traits. And we especially are applying a lot of next-generation sequencing and high-density genome mapping in order to improve our understanding of the genomes and the traits which are controlled by those genomes. And more recently, and really where we're heading, is trying to apply genome-based selection methods so that we can use these really high powerful genome data collection methods in order to accurately predict plant performance within breeding programs without having to phenotype every single line in multiple environments, multiple years, which is an extremely expensive enterprise. So this is the simple model of trait expression, which I'm sure everyone's familiar with. Basically, the fact that genotype and environmental variants are contributing to phenotypic variance, so that the trait expression is a product of this interaction between genotypes and environment. And what we do is to try and come at this from different levels to try and dissect this extremely complex interaction into different components. So we especially look at this on a population level. We tend to try and develop immortal populations which we can really phenotype and look at in great detail in order to extract environment effects from genotype effects. We do an awful lot of phenotyping on many different levels in order to try and look at interactions between traits and which traits are most influential in terms of the ones that we're really in the end trying to select for. Perhaps to find surrogate traits which can be used to simply select at least germplasm which is going to perform how we want it to. Then of course looking at the genome level, looking at genome sequences, genome-wide SNP profiles, for example. And beyond that, when we want to look at interactions between genes and epistatic effects, then really looking at the next level beyond the genome of expression. So we're looking at gene expressions and also metabolite expressions, or the products of all of those interactions between genes. Just one example to show the kind of population development that we're interested in. We spend a lot of time over the last decade or 15 years developing, for example, a big Brassica B.napus, an oilseed rape canola diversity collection of highly inbred lines, so they're homozygous lines which are then more or less immortal. And this population of around 500 genotypes has been distributed to people all over the world, in the hope that these people will then be doing interesting phenotyping studies, and that we'll get some of this data back and be able to put multiple layers of phenotype information onto each of the data sets for each of these genotypes. And at the same time, we've also genotyped this whole collection with genome-wide SNP markers in order to do genome-wide association studies and this kind of thing with the phenotype data that we're getting back. And we've been doing a lot of phenotyping ourselves also in collaboration with breeding partners and other institutes in order to really build up a basis of data for this immortal population. So we can do QTL analysis, classical QTL analysis, we can do metabolite QTL, expression, gene expression QTL, or with this kind of population, a genome-wide association study. In a similar way, we've developed for canola, in this case winter canola, a really powerful population over the last 5 years, which we're going to be able to use for nested association mapping. I'm not sure if you're all familiar with this concept of nested association mapping. This was developed in maize by Ed Buckler's group at Cornell, and we've basically reproduced this in canola. We've got a large collection of 30 natural winter-type Brassica B.napus accessions and 20 extremely divergent resynthesized accessions. So these are crosses between cabbage and oil, what do you call it in English, oil turnip, which is basically, these are the predecessors of Brassica napus, and we can reconstruct this, this cross in the lab in order to have extremely divergent inbred lines. All of these have been crossed with a common recurrent parent. In one case we've done back crossing in order to improve the performance of these. And these populations, or from each of these crosses we've got 50 offspring. So it's a total population of 2,500, just over 2,500 nested association mapping lines. Because we sequence the complete genomes of each of these 51 founder parents, we're able, using genotype data from a 60,000 SNP genotyping array, we're able to go back, and for each one of these 2,500 lines we can basically reconstruct the whole genome sequence of this line by looking at the recombination events and the genome sequences of the two parents that it derived from. So it's an extremely powerful collection for association mapping and really for gene mapping and understanding traits in this particular crop. At the same time, we've also taken 1,000 of these lines so far, the rest will be done later, and they've been used to make F1 hybrids so that we can also look at hybrid performance and heterosis by phenotyping the hybrids and then relating the data that we get here back to the genotype data and the whole genome sequences. So it's an extremely powerful resource which we think will be useful for genetics over the next 20 or 30 years, and is really as an immortal population, something which is extremely valuable. And we're working with the breeding companies who are involved to try and release this on a public basis so that people can get this with an MTA, to do work with it, with the assumption that some of the data that you will be generating will be flowing back into the producers of this data, or of this resource, so that there's a benefit, a mutual benefit for everyone involved. We're on a smaller scale. We've been doing similar kinds of things in sorghum and in wheat and barley also, where we've developed diversity collections together with breeders and different related mapping populations that we can use for genetic analysis and also for breeding. This is just an example from my postdoc, Liebig Bekele, who's here in the audience, who developed, based on some genome sequence data, a genome-wide SNP array, and has used it to genotype a number of populations that we're working with in sorghum. For various reasons, we've been doing phenotyping on these as well. So with these perpetual populations, not only the ones I talked about, but also just standard mapping populations, so double haploid or recombinant inbred line populations, we basically take the approach that these things are immortal resources. We have seeds which we can reproduce every year or every couple of years. And every piece of phenotype data that we get for them can be put into a database and compared with other phenotypes for this particular individual. So we put these into multi-location, multi-year field evaluations, and we look at all of the kinds of things that a breeder would be interested in the field. But we also put them into controlled phenotyping. So, for example, we can do stress phenotyping and look at things in a very controlled manner to reduce the environmental influence. To look at, for example, germination, emergence, we've done a lot of work recently looking at root traits, abiotic stress or nutrient responses, and so on. We can also do metabolite analyses or transcriptome analyses in these controlled conditions in order to give us really a number of different layers of information about a specific genotype and about the population that it belongs to. And this is basically what I'm going to talk about for the rest of my talk, just to give you an example of how we approach this. And the example is something which is a little bit more complex than a normal genotype-environment interaction. We're looking at emergence, or germination and emergence as a complex. And this is something which is it's more complex because we don't just have a single environment that we have to deal with. We have, of course, the environment where a seed is sown. So you have the soil environment, soil moisture, encrustation of the soil surface, temperature, and so on. And of course, all of these things will influence the way that the seed germinates and begins to grow or doesn't. But also, then you have the environment where the plant is harvested. So this can also be influenced by temperature, by nutrients, by water status, and so on. And all of these things influence the quality of the seed. So the quality of the seed also influences how it germinates. Okay. Again, then you have a storage environment. So depending on the quality of the seed, you may have a different capacity to be stored for a certain amount of time. The temperature and the conditions at which you store also influence its capacity to germinate and so on. And I think you're getting the idea. All of these things interact and give you an extremely complex genotype by environment interaction which influences the way that any given seed will germinate in any given environment. Okay? So it's much more complex than the general picture that we're normally looking at, and it makes it really difficult for a breeder to select based on a phenotype whether a particular genotype or breeding line has a good germination and emergence or not, okay? Because he has to take all of these things into account, he or she. So we're looking at this complex of traits, especially in sorghum and in canola for different reasons. In sorghum, because the cold temperature emergence or cool temperature emergence in springtime in Europe is really the do-or-die trait. If you don't have it, then you won't be able to sell seeds. So really we're interested in this as a way to establish sorghum as a new bioenergy crop for Europe. And we're also interested in it in canola for various other reasons, but I'll come back to those later. So in sorghum, as I said, we basically have these different kinds of populations, and we try and phenotype them for anything we can think of, which may or may not be vaguely related to germination and emergence under cold temperatures. And so we did an awful lot of phenotyping with many students over the years, looking at germination and under different temperature constraints, water constraints, and so on. Growth chamber testing, greenhouse testing, as you can see here, and really with all sorts of different stress applications, just repeating experiments over and over and over again to give us as many layers of information about these populations as possible. And what we end up with is an awful lot of phenotypes, and we can look at the phenotypes in terms of correlations. So we can make a correlation matrix which shows us how specific traits are interacting with others. And you can see these red or blue boxes here show that there's a positive or negative correlation between traits. Okay? So as you expect, it's pretty complex, but we can also put all of these things onto a genome map and try and look at QTL or quantitative trait loci which are underlying these traits. In canola we're doing more or less the same thing, perhaps on a slightly larger scale and with more field data. In this case it's really about establishment before winter, because we sow our canola in August, so around about this time of year, and it'll establish in the next few months if it's not too dry or too cool or whatever. And it really has to have an extremely good establishment in order to survive the winter which is coming. Okay, so it'll grow through the winter and flower in the springtime. And the winter in Germany, of course, can be rather harsh. And in fact, canola is one of the crops, at least in Europe, which has the most erratic emergence during this time of year. It's really about the sowing time. There can be a very big fluctuation in the temperature and moisture conditions of the soil. So we have a big fluctuation in the quality of seed, and also we have a big variation in the ability of a seed to germinate depending on where it was grown. So there's this other environmental effect also. So the breeders are extremely interested in trying to find ways to improve this trait. So what we've done here, again with these really diverse populations and well-characterized populations, we're trying to phenotype anything we can think of which may be of interest with this emergence trait. So we've been doing a lot of controlled environment phenotyping, some very simple gel-based germination and growth assays, more complex lemnitec phenotyping here, for example. In this case, we're looking at drought resistance traits. Here, in vitro phenotyping with a simple PEG assay to look at osmotic adjustment and so on. On the bottom right here, you see a nice system we've established with these, we call them the Giessen large container and phenotyping system or something. It's just like a whole lot of rubbish bins where we're growing canola under a rainout shelter. So it's actually a really nice system for drought phenotyping, for nitrogen use efficiency phenotyping, because we can grow the plants at more or less a field density, and they have sort of the interactions that they'd have in the field, which for canola is an extremely important thing. It's impossible to get a yield-relevant result from a single canola plant in a greenhouse. It just doesn't work. But we've found here that we can get correlations of around 80% to field yields, which is really quite astounding. So this is a nice system also for all sorts of other things, like metabolite analysis and transcriptome analysis. We can use this controlled container condition. So we control exactly the amount of water and nutrients that are going into every one of these containers. In terms of emergence and germination and emergence, we've been working very closely with the French seed testing lab in Angers at an organization called GEVES. It's the National Seed Testing Organization. And they've got an exceptional system where they'll put seeds onto these plates and visualize their germination rates and the growth rate during the first 72 hours of after germination. And they can put through around 2,000 genotypes a week, so it's really a high-throughput phenotyping system. We've used it to phenotype many of our populations. And here you can see one of the things that I was talking about, that with the same population, here we have a seed production from France in 2011 and another one from Germany in 2012 with the same population. So this is a diversity set. And you can see that the rate of germination changes considerably between these years. So here the seed quality is obviously not as good as in 2012. But also the extremes show a much more extreme reaction in some, if they're being produced in some environments. So this is not the environment in which they're being sown. It's the environment from which the genotypes came. And at the same time, you also have this variation in the environment in which they're being sown. So there's a genotype effect here. So we can simulate this, for example. We can change the water capacity, for example, and simulate a kind of a dry soil germination. And here you see the change to a very, very delayed germination in some of these genotypes. So we can put extreme genotypes through these different environmental constraints and see how they react. So that we basically build up for each of these genotypes a catalog of information about how it copes with different environmental variation. We can do the same thing with temperature. As you can see here, of course, as you'd expect, by increasing the temperature, we can also increase the growth rate. But again, we get big variation in the phenotype. So what we've done with all of this information is to try and then compare it to field data, because the breeder is interested in trying to find out what can he look at in one of these high-throughput phenotyping platforms, which is going to give him an idea of what he really wants to be selecting for. So we've had a group of breeders working on this project who have phenotyped the same populations at, I think it was at that time, 9 different locations over 2 years, so a total of 18 different environments. And they've looked at many different traits which a breeder would normally go out and screen in an extremely labor-intensive process. For example, the plant biomass after 2 weeks or 4 weeks or something after sowing. And so you can see here all of the automated phenotypes that we collected on the right and at the bottom. And on the top at the left, the average phenotype data that the breeders were generating from all of these locations. And what we do is to try and find the one or the position where a high throughput phenotype is relating to something which the breeder wants, or is correlating to something which the breeder wants to select for. And we've got one good hit here where we have a very good correlation between what the machine was scoring and what the breeder is scoring. And so we can use this particular phenotype to go to other populations and to screen and really look at the genetics of this particular phenotype in order to understand it better, in order to really eliminate the environmental variation that we normally have to deal with in such studies. Coming back to sorghum, just as an example, what we then do with this kind of data, we can put it on a genome map. In this case, it's the 10 chromosomes of sorghum. And this is, again, some work that Liebig has done. And you can see here, the concentric circles, like the wheel of this diagram, are the chromosomes, and these colored bars are basically QTL for all of the different things that we've been phenotyping. So the regions on the chromosomes where there's an effect on a particular trait. And it's an extremely complex diagram that you can see. You can see in the middle all of the gene-for-gene or the locus-for-locus epistatic interactions between different regions of the genome which are interacting to also influence particular traits. And it's extremely complex, but at least we can focus in on specific areas where we have like a concentration of QTL, for example, for many different traits. And we can say, okay, this seems to be a region which may be like a major or a master regulatory region for all of these different stress-associated traits. And we can try and focus in on these by looking at a higher density on the genome, using a SNP array, for example, as shown here on the left, or by using other materials, other populations to fine map this kind of region. Just as an example, if we go from this biparental QTL analysis and try and focus in on interesting parts of the genome with a genome-wide association approach, then we can also see for particular traits, this isn't working anymore, we can get significant genome-wide association study hits, which point us to really sometimes quite narrow regions of the genome where we can look for genes that may be involved in, or may be candidates for the traits of interest, and may be interesting for breeders. In this case, for example, just taking one of these interesting QTL that we found in a biparental cross, we can also map the same trait or a related trait in a diversity panel, and we can get a relatively high resolution. This is not a really high density SNP array that we've used, but we get a pretty high density resolution which narrows it down to a few genes, in this case 26 genes, including a homologue of a gene which we might think could be involved in a trait like cold tolerance or even a general abiotic stress response. And we're currently just reanalyzing all of this data with a much more high density SNP array with 90,000 SNPs in order to try and saturate particularly the QTL regions that we're most interested in, to try and really identify the genes of interest in these. A high-density SNP array for canola is already available. Dave and Jackie were also involved in developing this array. And this has been really, really powerful for genome-wide association studies in canola. There are a couple of examples which I could show. I've just pulled one out. This is an analysis of seedling metabolites, which are implicated in this early germination and especially vigor response. And what we've found here, in one case, for example, at the bottom, a very significant hit with which is very close to a gene involved in sodium exclusion, for example. So it's giving us an idea that some of the emergence and vigor traits are actually related or relating back to stress tolerance, even though in this case we weren't phenotyping a stress tolerance trait. Okay. Another thing that we've been doing with quite a lot of success has been genome-wide sequencing in order to narrow down QTL regions. And here, using a kind of a bulk segregant approach, we've taken a group of individuals from a segregating population which have a very poor chilling survival in sorghum, and another group which have a very good chilling survival, and basically done a whole genome sequence of each of these recombinant inbred lines. And we then can scan across the genome using a kind of a sliding window approach to find the regions where the allele frequencies within these two pools of the good and the poor performers, where the allele frequencies diverge the most. And these are the regions where what you would expect to find would be that the allele frequencies in these two pools would be more or less constant over the whole genome, unless you come to a genome region which is segregating because of this particular trait. And so where the allele frequencies diverge the most, these are the points where we expect to have the QTL and genes involved in these particular traits. And this is just one example. You can see here we have one point where the blue line for one of the pools is way at the top, and at the other pool way at the bottom, and the red line the other way around, for example. Okay? So this is the kind of thing we're looking for, and you can see here we have, it's not a simple trait, so it's not a single QTL where we're mapping a QTL as you often see in the literature where people have used genotyping by sequencing to sequence single QTL. This is doing it for multiple QTL, so we're not getting complete hits. These QTL aren't explaining all of the variation, but we're getting at least a much closer or a much more accurate delineation of the QTL confidence intervals. We can also use this kind of data. So this is simply the genome sequence data from these inbred lines which we used for the previous diagram. And we can use this to map the recombination breakpoints on every chromosome in every line. And this data we can use for high-density mapping. So we can generate a recombination breakpoint map, in this case, which is enriched for markers or polymorphisms which are near to QTL, because we've selected the individuals based on the trait of interest. So we have a lot of recombinations at the positions where we're looking to map the QTL, and this helps us then to get a very, very high density or high resolution QTL mapping for particularly the traits which we're interested in. And here again, we've mapped this to a very small interval with a relatively small number of genes that we can then go on and look in more detail. And of course, we have the genome sequences of all of these genes from the parents as well. So we can look for functional polymorphisms and so on and try and select candidates that may be interesting. Okay, what do we do when we have this kind of information? And this is what I'm going to finish with. This is just like a functional mapping kind of study. So it's really giving us no further information. And if we have candidates, it doesn't really tell us these are really the genes that we're interested in. So you could go back and do reverse genetics approaches and try and find out if some of these candidates really are doing what you expect them to. But in some of the crops like winter canola that we are working with, it's an extremely long process to do reverse genetics. And so we prefer to try and find other levels of information which may support our selection of candidates and genes. And the thing which has worked very well for us so far is a transcriptomic network analysis. Here, using canola as an example, we have been doing an RNA-seq-based population genetics. So it's going into groups of individuals which segregate for the trait of interest and doing kind of bulk segregant transcriptomics. So in this case, we take canola plants and we've got a lot of field data, so we can select accessions which are very, very poor or very, very good under field conditions in terms of their emergence and or germination and emergence. And then we can take these same cultivars and put them into an extremely artificial system. So in this case, it's an in vitro cultivation, so basically a hydroponic cultivation, where we grow the things for about 3 or 4 weeks, and then we can sample root and shoot tissues, and it's extremely clean material for transcriptomic and metabolomic analysis. And we use these, for exam' here for a 3' EST profiling, so a quantitative analysis of the whole transcriptome. And when we get this data, so we get transcripts, for example, from a shoot sample from around about 30 or 40,000 different canola genes. So there are 110,000 genes, I think, in canola have been found. And we're picking up around a quarter of them or a third of them in these shoot samples, which doesn't help us terribly much. So we first begin by placing these into groups of genes which show some kind of co-expression. It's simply a kind of a tree analysis where we can cluster genes based on co-expression. And these clusters, we then give a color, and you'll see these colors on the following diagram, because we can use these basically colors to identify a group of genes which are co-expressed. And normally these are genes which are belonging to the same biological function. They may be in the same pathway, or they may be expressed under certain conditions or so on. And what we're interested in is whether any of these gene expression clusters correlate also to the traits of interest that we are looking at. So what we've done here, it's a simple Excel diagram where the fields in the boxes in the Excel diagram have been overlaid with a color which shows the correlation between each of these expression modules, which were represented by the colors on the previous diagram. And on the bottom we have, or on the x-axis, we have the traits that we've looked at. So we have these high-throughput automated traits, and here all of the traits that have been looked at in the field. So each of these boxes is one trait in one field environment in 1 year. Okay? And if you have a dark red box, then this gene expression module is showing a strong positive correlation to this trait. If it's a dark blue box, then this expression module is showing a strong negative correlation to this particular trait. So we're interested in gene expression modules or groups of genes which are co-expressed and which show a strong positive or negative correlation to as many of these field traits as possible. These are the field traits which the breeder wants to select. And you can see very easily in this particular sample from 28-day roots that there's one module here which is really associating with many, many different environments, many different traits. So this is something which is probably relevant for a breeder. It's something that he may even be able to use to select a high or a low germination and emergence line. We can do the same thing in shoot samples, and here again we get a group of genes, one module which has a very strong correlation to these traits of interest. Interestingly, if you look on the left here to the germination traits, there's one box which is dark red. This is the one which I said before has a strong correlation to the field traits. In this case, we have a negative correlation to the gene expression, but it's again confirming that this particular module is something of interest to us. So without going into any detail on the bioinformatics behind this, what we do is we take the genes, all of the genes in this particular gene expression module, and we do what's called a weighted gene network co-expression analysis, which basically looks at the connectivity among each of the genes within that module, whether a gene has a correlated expression to any other gene in that module. Okay? And we get this kind of diagram, which basically shows which genes are the most connected to all of the others. And these you expect to be the transcription factors or the driving genes which are influencing this whole expression module. And there may be one, there may be many of these which are co-expressing. What we're interested in then doing is ranking them according to their interconnectivity. And we can pick out the ones which have the highest influence on this particular network as the potential drivers of this network. Okay? And interestingly, in both of these cases, both for the root and for the shoot expressed genes, we find a single gene which has a 99% correlation to the traits that we're interested in. And the next best hit has around about a 95% correlation. So we think that the one with 99 probably is something quite interesting. And they seem to also be the kind of gene which we'd be interested in looking at. So one of them is involved in root hair elongation, which is kind of a root trait which is perhaps going to help a plant to take up nutrients and so on during its early life, during its emergence. The other one is a gibberellin-related regulated protein, which is especially involved in gibberellin stimulus responses. Okay. Just to go back to one other example where we can find interesting information from this kind of data, if we look at the bottom here on the shoot expression, so the one at the top is the one we just looked at. If we look at the bottom, you can see this one module where we have some interesting correlations with expression. But if you match these up to the top, these are environments where we have a switch in gene expression from the top module, which is now showing up as a blank, to the bottom. So it's a different regulation depending on a specific environment. And so we think that this may be giving us some clues about expression of traits in specific environments. So perhaps QTL or environment-specific QTL. If we take, this is something which we actually do for all of the modules we're interested in. We would like to map the genes involved in these modules, especially the ones with the high interconnectivity, to QTL, which we've established for all of the traits that we've been looking at. And if we take this particular example, we take these, all of the genes in this yellow module at the bottom here, and we map them onto a QTL map where we've done genome-wide association studies or biparental QTL mapping to find QTL influencing traits of interest. And this is really interesting because we find 2 transcription factors in this particular yellow module, which both map within a biparental QTL, which is also a GWAS QTL. So it's a QTL which is relevant in a very diverse material. But this particular QTL doesn't always show up. It's an environmentally specific QTL. So what we're now doing, now that we know that these QTL are somehow environmentally specific, and they're also we know some of the genes which seem to be involved in regulating these QTL. So we can go back to environment data, and we can say, what was the soil temperature, what was the moisture content of the soil, what was the outside temperature, how much rainfall was there, and so on. And we can try and put this environmental data back into a model which is going, including genetic, transcriptomic data. And we think that this may be a key to helping us to predict performance of a given genotype in any environment where a farmer may want to grow that seed. And this is something that we're just beginning to work on, trying to integrate climate data with modeling for trait prediction in order to help breeders to really give farmers advised choices about what seeds they should be buying for what particular environment. So finally, this is kind of where we're trying to head. We're collecting an awful lot of data from many different perspectives, many different levels of data, which is somehow related to these complex traits that the breeders are interested in. And we think that all of these different kinds of data will help us to somehow model performance in order to develop prediction models that we can then apply in really commercial breeding programs. Basically, it's a description of genomic selection, if you're familiar with the way that we try and use genome data to predict performance. And we think that all of these different levels of data will really help us to improve genomic prediction models and help breeders in the future to make more resilient and more environmentally adapted crops. And I think that's probably a good place to end. So I'd like to thank you all for your attention. I have one last slide where I've named a number of different cooperation partners throughout the world and throughout Germany who we've been working with on a scientific level over the last decade or so and are continuing to work with. Many thanks again.